On your New England rare plant reporting forms, you will see two different ways of reporting the number of plants you find at an occurrence. The first one asks how many ramets are present. A ramet is a vegetatively produced unit of a genetic individual, for example, a stem. Oftentimes, your first count of plants will be of ramets, how many stems you see of that species at a site. Both of the drawings below show three ramets, or three above-ground stems, all connected back to distinct genetic individuals. You will also see the term genet, which is a unit of a population arising from seed, not vegetatively produced from another individual, genetically distinct from other plants in a population. This means the whole plant, the entire genetic individual. Some species send out underground runner stems that produce multiple ramets, but are really one genetic individual. It is helpful to get a sense of your species biology. Does it produce underground stems, grow in a clump, annual, or perennial? Before going out on your survey to get a better idea of how to count the plants you find. We understand that it can be hard to determine the number of ramets and genets when you cannot see what's happening underground. Make sure to research your species and give us your best estimate when in the field.
Here is another simple drawing of some plants. Can you tell how many ramets there are and how many genets there are? There are 5 above ground stems, so there are 5 ramets. Thanks to our X-ray vision, we can tell that these stems arise from 3 distinct plants. Now, since we do not dig up rare plants, unless you are a superhero, you will not be able to tell what is going on underground, so it is crucial that you familiarize yourself with the growth and dispersal patterns of your target plant, so you can tell the difference between ramets and genets out in the field. Sometimes when you get to your survey site, you will find a relatively small population, anywhere from a single plant to 50 plants, which is easy to count and track. This is a photo of a population of large whorled pogonia, Isotria verticillata, and as the arrows indicate, you can clearly see 3 individual plants in this photo. Large populations of rare plants are a little trickier, and you may have to estimate the count of plants in a large population. We require all surveyors to report a whole number, either exact or estimated, of either ramets or genets for a found population. One method to go about counting and estimating the size of a large population of plants is to first count how many plants are in a square meter, then multiply that by the total square meter area occupied by the entire population. Make sure you count plants in an area of average plant density, not on the edge where plants are sparse, or in a patch of densely packed individuals. This photo is a good example of a very small rare plant. Small flowered dwarf bulrush, Lipocarpa micrantha, in a very large population. This tiny plant is only a few centimeters wide, and in the second photo, a volunteer is squatting surrounded by a giant patch of this pond shore tiny plant. It would be too difficult to count each plant individually, so an estimate is best for everyone's sanity.